Assalamu alaikum wa rahmatullahi wa barakatuh, and welcome to this after show. Today, unfortunately, Dr. Fazrana had to catch a flight, so I'll be doing this today alone, answering your questions if there are any about our episode, or actually, since we're here together, about any of the other topics that we have discussed in evolution, or even some topics that you would like to open. So I'm glad to receive your questions over the live chat, and we can just have a nice discussion together. So let's see who will give us the first question now. So as you have seen today, it was about the DNA storage, and I have been personally fascinated with this topic, where we have seen together that DNA is not just an information molecule, not any information molecule, not just some chemical compound where a specific arrangement of nucleotides can reflect bits, but that the very structure of DNA lends itself to DNA being an information storing molecule. It's completely amazing that this is the case. And what it says is that there has to be, it is very logical and it is very natural to think that there is a designer behind it, because you cannot just expect and you cannot just accept that DNA just looks like this just because. DNA is in the way it is because it is designed to be this way. And it is because of this design that there are properties that DNA has that are very suitable for DNA being an information bearing molecule. And when you see the purpose that the DNA serves in the cell and the design features that DNA has, then it must be the case that DNA is designed by a designer, and that this designer, who is God, obviously, has made it this way because the fulfillment of the function came first. So there is purpose, there is intent, there is design that fulfills the purpose, and then there is essentially the implementation of the design, which is DNA itself, which can now naturally and logically exhibit those features that we have seen. So let's see if any of you has questions, or we'll just call it a day. And I would have just liked that Faz would be with me here today, but since he's not, then if there are no further questions, then we can just close the live stream, and I will see you again next Thursday, inshallah. All right. So, all right. So here is a question from our friend Yoshi. He says, "I agree with all that you say. I might ask something." All right. Yeah. Sure. Go ahead. Yeah. That's why we're here today. So if you have a question, just go ahead and ask. Yeah. What is it? Mm-hmm. Okay. What's the question? Okay. So he says, does duplication add new DNA code to the existing DNA code? Let's put it on the screen. Okay, so there is this process where there are some elements of DNA called transposons, which can just jump from one place to another and even create duplicates. And there is a process of creating duplicates of the existing DNA. So does this create new code? It actually creates a copy of the very gene that has been duplicated. So this is what happens when this process occurs. Is this new code? It is essentially a copy of the existing code. But sometimes this process of duplication or of transposition, it can cause that some functions that were not possible before the transposition become possible after the transposition or after the duplication, because the new location will enable something. So, for example, if you have watched the interview that I had with Dr. Michael Behe a few days ago, which is this one, and I hope if you haven't that you would actually do, because it was really a blast. So if you watch that, we spent some good time talking about the Lenski long-term evolution experiment. And in this experiment, there are at least two categories of things that happened that were observed by Richard Lenski. One of them involved a breaking of a function, which caused the bacteria to become larger because they wouldn't stop growth at the typical limits. And another one involved that the bacteria started consuming or metabolizing an energy source called citrate, and that happened when a piece of its DNA that used to be in a place that is inaccessible, when glucose was not in the environment, that piece moved to a different place, which is after a promoter, that hence causes this piece of the code to be read. Okay, and when this piece of the code is read, then this piece of code was coding for transport proteins that would cause citrate that is in the environment to be able to come through the cell membrane and be transported inside the cell, and hence metabolism will start. So in this case, an already existing piece of code came to a new place right after the promoter that would cause the transcription of what comes after it. And this new addition of that code caused the transporter protein that is produced to be able to transport citrate. And this is obviously a reactive adaptive response from the E. coli, which is the kind of bacteria that was in this experiment, because it needed to consume citrate. All the machinery of the cell existed for the consumption of citrate, except that the transport path was not open because the transporters were not transcribed. And in this case, the duplication can actually, or the transposition, which is the movement of a piece of a DNA from one location to another, caused an enablement of a function. Now, this is one thing, and how evolutionists address this duplication of DNA is a completely different thing. So what they say is that you would look at earlier creatures and they have limited genomes, and you look at later creatures and it has a much bigger genome. For example, it is postulated or proposed that Luca, which is the last universal common ancestor of life, had 350 give or take genes, and humans have 25,000 genes. So then you will have to ask the question, where did the extra genes come from? And then they would tell you, yes, it is gene duplication. But then when you tell them gene duplication will essentially just copy all the existing genes, so making a second or a third or a fourth set of the genes wouldn't actually create any new functions, they would tell you yes, but then those new genes will be left in places in the genome, and then that those genes, because they are not important, because they are not needed, they will incur mutations. So mutations will distort those genes, and then those mutations can cause the information stored on the genes to change, And then at some point in the future, maybe through the so many mutations that happen to the genes, that those genes will now become producing proteins that make sense or that produce a function. Obviously, this is a nonsense argument, in my opinion, because it is. I have a little video that talks about this kind of nonsense argument. I give this parable. There are so many. Number one, there are so many molecular reasons why this will never work, including, for example, that these genes that are duplicated, there are some areas that are sensitive in those genes, like the promoter areas, etc. And when those are damaged by mutations, then forget it; they are dead. But then again, let's imagine that we have a program, and the program copies itself. Okay, you can allow that the program will create of itself various copies of parts of it, and those parts will remain unattended in the computer memory. And then those parts, since they are not important, they can get scrambled by different electronic events that nobody cares about. So somebody writes on top of them glitches of electricity or glitches to magnetism cause the changes of the bits or whatever. Do you expect that those parts that are sitting in not important places in the memory, after some time will produce new functions or new procedures or new modules, okay, that will deliver extra useful functionality to the original software? Not only that, but that the original software will find it, will call the appropriate functions, and that this thing will look to the user as a harmonious creature. And obviously, if anybody knows ABC of programming, he will immediately know that what this fantasy that I'm talking about is nothing but a fantasy, not even good for a science fiction program, and that this is totally something that will produce just rubbish code, not something new. So the whole argument of duplication of DNA, etc., I would say can fall into one of two contexts. The first context is that the code is actually managed by the pre-programming of the cell to be activated or moved to a different place to produce a certain response to a certain environmental condition, like, for example, what we have seen, whether it just moves, completely jumps, or gets duplicated, like what we have seen in the Lenski experiment. Or this is a duplication that creates a duplicate of the same gene, which will then, for example, cause the same gene to be transcribed double the time, which means that whatever this gene was producing will now be produced double. Or that this event will be eliminated by the genetic system because it was not needed, and it will just go away. But to assume the impossible scenario that this duplication will cause novel information through completely blind mutation is, in my opinion, nonsense. And there is no empirical evidence for it, of course, like any other one more of those crazy suppositions that evolutionists will put. So we have here Salah ad-Din saying, alaykum as-salam wa rahmatullah. And then Yoshi says, so duplication can increase DNA in volume even though it doesn't help evolution in the slightest. Yes, it can. But I just would like you to take this in a grain of salt, because what we know about DNA is so small. Okay, we do not really understand. So this example that I have told you about, the Lenski experiment, is an experiment that happened over 30 years. Okay, and then because there were very controlled conditions, so exactly what Lenski was doing to the environment of the bacteria was very controlled. He was increasing food, he was removing one source of food, he was adding another source of food, etc. And he was doing this and monitoring exactly what's happening. So he managed to capture a specific event where he saw that the bacteria is now for example, growing larger or now metabolizing citrate, and then he would take that sample and sequence it and see what happened. And then we know that this kind of transposition can cause that thing, and that this thing is obviously very related to what has happened in the environment. It's an adaptive response. It's a pre-programmed response. Now, what else does the cells use gene duplication for? We have no idea. But what we know, for example, is that the human genome is like 3 billion base pairs. We only understand what 1.5% of it is doing. And one of the propositions was that the rest was completely junk, and the rest was completely produced by duplication events and collection of different kinds of rubbish. But then a project was launched, and a criteria was set that DNA is junk if it's not transcribed, because if it's not transcribed, it means that the cell does not use it for anything. Then it means it's just complete rubbish or junk. So the term junk DNA. But then the surprise was that the ENCODE project, which was an international effort, found out that like 80% of the human DNA is transcribed. Now we also have now there is another problem, is that because we have no idea what it's transcribed for. So it's transcribed and it produces something, but we don't know what this thing does. So if you just think of a mental image of what I am telling you, we only understand what the 1.5% of our own genome is for. It produces those kinds of proteins, for example, and we don't really even understand what this 1.5% exactly does. We know that this gene produces that protein, but what that exact protein does, if we did understand, then probably would have been in a much more advanced status in medicine and treatment of cancers and many other things. So even the 1.5% we don't fully understand, and nobody claims that we fully understand, and the 98.5% we don't understand at all. So now microbiology and evolutionary biology is in a problem, because this 98% seems to be transcribed, and hence, since the cell is consuming energy to transcribe it, because it does not only have it, it's replicating it all the time, it's transcribing it. It means it's converting it to RNA, and then this RNA might be used for something, for regulation or for producing of other products, etc. But we don't understand what it's doing. Really, I think that this matter of duplication, why the system does it, what it's used for, will be something that will be under further research for the coming few decades. Okay. So Salahuddin says, okay. Yeah, so he says again, so duplication can increase DNA volume, even though it doesn't help evolution at the slightest. Yeah, it can increase it in volume, but as I explained to you, sometimes the transposition is just moving a piece from one place to another. Sometimes it's duplicating. So it can increase it in volume, and I don't think it helps evolution in the Darwinian sense. Yeah. He then says, I agree that function can be created only by mind, because function is when an object has meaning, and meaning is information, and information can be established only by mind. Yes, I agree to that, definitely. Yeah, and then he says, Salahuddin, it seems that Ahmed is giving the answer yes, even though I agree that it doesn't make Darwinism a more intellectually satisfying model. I agree to that. All right, so okay, so that was about this matter of duplication. So do we have any further questions? Thank you. So Yoshi says, you've explained it brilliantly. Thank you. All right. So that was about this matter of duplication. We will also, by the way, in this series with Faz, whom I have explained earlier that he had to catch a flight, so he couldn't do the after show today. But we will be also addressing some very interesting topics like pseudogenes, etc., which are related to other topics like ERVs, etc., later in this series. And to me, what happens is that we have this, ironically, evolution of the gaps issue. Okay, so evolutionists do not understand, evolutionary biologists and microbiologists do not understand so much, and then they try to fill the gaps of misunderstanding by those premises and presuppositions of randomness and the Darwinian mechanism filling these gaps to create things from nothing, or to create order from chaos or design from randomness. And it occurs that as science progresses, that those areas that they are abusing to put further there or to continue propelling their arguments is shrinking with time. Yeah. Okay. So Yoshi says I might have new questions. Go ahead. Yeah, go ahead. If you have another question, go ahead. Whether it's about duplication or anything else, I'm fine. Yeah, it is atheist, God of the gaps, as Salahuddin says. I agree to that. It's yeah, they don't have a god. It's just the atheists' arguments of the gaps, or evolution, or Darwinism of the gaps, if you would like. Yoshi says, but cell is a finite space, and duplication cannot increase DNA infinitely in it. Well, yeah. So that's what I was saying earlier. So we see that there are duplication events. Okay. Now, are they going to drive the genome to infinity? Obviously not. We don't have creatures that have infinite genomes. And let me tell you this process is definitely a regulated process, because and those duplications and whatever happens after them is managed definitely by mechanisms that we don't understand. And to give you some evidence to that, let me tell you this. For DNA to just get duplicated and move to somewhere else, there needs to be a specific kind of code attached to that DNA that enables that piece of the DNA to duplicate and reinsert itself some other place. But we also know that there are parts of the genetic system that can inhibit completely parts of the DNA from being transcribed or for being read altogether. So I'll give you an example. Your brain and your liver look completely different. We all know that. And your muscles and your bones and your blood vessels, all of those tissues that are parts of organs, and your skin have cells that look completely different from each other and that have different functionality and property and proteins, etc. But the surprise is that every one of them has exactly the same genome. So you have the same 3 billion base pairs all over your body. What makes them look differently is that only a specific part of that genome is enabled for transcription in every kind of cell. Okay, so there is the epigenetic system, and there's the histones and the methylations, etc., that inhibit the DNA from being like randomly everything is getting transcribed all the time. And if you go to the On Evolution course, to the Central Dogma episodes, which are the last 3 episodes, you will find me talking about that extensively. And it's totally and completely amazing. So those transposables, or those pieces of the DNA that have the ability to jump from one place to another or to get duplicated, need to be from the very beginning enabled to do so by the epigenetic system. So the system itself enables them to do that. And when enabling them to do that, they would then move further to do the rest of the cascade of the events that are dependent on them moving from one place to another or getting duplicated, like what we have explained in the Lenski experiment. Now we have one of two scenarios. Scenario one is that this is an intentional thing. So now, because of this transposition, you get a new strain of the E. coli in the Lenski experiment that has the ability to metabolize citrate completely. Not only the 90% of the machinery that can metabolize citrate is there, but also the final building block of enabling citrate to come in is enabled in that strain that had the transposition happen. Then this new modified organism will strive better in environments where citrate exists. Okay. Now, the other thing is the evolutionary idea that this piece of DNA is just jumping around and getting duplicated randomly without any purpose. Okay. So if this is the case, and if we have a cell that has a crazy duplication function that keeps duplicating, so you have the first original cell without the crazy duplications, and you have the second cell with the crazy duplications. And then the second cell will now have a larger genome that the extra parts or the new parts of the genome is not contributing any extra benefit to the cell. This means that the first original cell will now be more energy efficient than the second cell, because this one has a smaller genome, replication costs less energy, transcription costs less energy, it's carrying less baggage with it. So what will happen? It will happen that this one will be more fit than that one. Then the strain that has the nonsense duplications, the random duplications that come with no extra advantage, will not have an advantage versus the first one. So this one will prevail and this one will go away. So I guess having just said that, what it means is that if there is a runaway duplication process that will just continue just consuming energy for the duplications and then enlarging the genome for no reason, that this will be eliminated by fitness automatically. And that's why the duplication will not cause an infinitely long or infinitely large genome. And the evidence that we have that the genetic system is very sophisticated, everything is under control, everything is regulated through epigenetics and through very intricate mechanisms that also relate to the promoters and the transcription factors. And you can learn about those in the evolution course. It is episode 8, and you have 8 and 8 part 1, 8 part 2, 8 part 3. And you can look at that in detail, and we will also be addressing it here when we reach to the central dogma in this series with Dr. Faz Rana, Seeing God in Biology. You will see how intricate this system is. So it is very unlikely that these duplication processes will just happen and be runaway processes. And in case this happens, it's like a kind of disease or a kind of dysfunction, then it will be naturally ruled out by fitness. Because if you have two cells and one of them is less energy efficient, it just gets ruled out. So I hope I have answered your question in a satisfactory manner. And let's see if we have more questions today. Just until this happens, I will, until somebody puts a question, I will put on the screen one. Yeah, I think it was this one. This one will be beneficial for more the genetic code design on the evolution course. Is evolution true or false? This was part 2. I think it's interesting for you to go there and look at part 2 and even part 3, which is this one, which talks about from DNA to proteins, and it's completely mind-blowing how the system is sophisticated and how there is really great chains of events and specificity in everything in the genetic system, to the extent that it is impossible that it is actually things will just go randomly and just run away unaccounted for. All right, so so Yoshi says, thank you. I feel I'm taking too much of your time, but unfortunately I have some questions. Find out regarding the duplication. Okay, so let me have it. What's the next one? Let's have a new question from you today. So it's good to have questions, and inshallah other people will benefit from them. Okay, we have a question from Salahuddin also. He says, until Yoshi also brings forward a new question, he says, What's your comment on 98.7% genetic similarity between human and chimp? Okay, so this is a typical proposition from evolutionists, and there are the proposition is very flawed from many aspects, really. So let me tell you some of those aspects, and I would also just for you also to have some more fun with this, I would also suggest to you to see an episode of mine under Truth, I think, series is called Adam or Ape. It's a fun episode. So who Adam or Ape, who is our father? That was its title. If you go there, you also find that argument why there is no, at least there is no known path from apes, whatever they are, from ape ancestors to humans. But let me talk about this similarity issue. Number one, similarity is not an argument altogether, because similarity is a fact in living things. Okay, now whether similarity is 50% or 70 or 80 or 90% or 100% has no value, actually. Why? Let me tell you this. Number one, if you look at animals or if you look at vertebrates, let me give you an analogy. If you look at cars, and you want to write a book, okay, about everything that describes type 1 of a car, okay? Let's say this type 1 is a Toyota car, okay? So you write a book that describes everything for somebody who has no idea whatsoever about building a car. He can use this book to build the car from scratch, from basic materials, okay? To build the car from scratch. Because this is what the genetic system does. It builds a complete living organism from scratch, including converting essentially atoms or small molecules into larger molecules, into complexes, into, so this is what DNA is, the recipe for building the organism. So let's say we are now building this book, okay? We are writing it to give somebody the idea of how to create a Toyota car from basic materials. And then somebody else will have to write another book, okay, that will completely explain how to build a BMW from ground zero, from basic materials. Now, my question to you is, how much similarity can you expect between those two books? I would expect that the similarity will definitely be more than the 90%. Why? Because the book will have to have the recipe for how to create steel, the recipe for how to create glass, the recipe for how to create rubber, the recipe for how to create plastics, the recipe for how to create foam for the chairs, the recipe for how to create specific alloys that are used in the engine, the recipe for how to shape and how to make something that looks like pistons and tubes, etc., because you're building the whole supply chain and the whole bill of material of the car. So at the end of the day, when you look to the two books, you will find the part that addresses the raw materials and the part that addresses the many components and the many intermediate products and the many intermediate machines, etc., 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 will be very similar. Okay? And then what will be the real difference between the Toyota and the BMW? Maybe the assembly instruction, maybe the level of refinement of some of the parts, but even those assembly instructions, you will need to do the assembly. There will be plenty of commonality there. You will put the shaft, and then you will put the the axes and you will put the wheels, you assemble them in this way, etc., etc., etc. So at the end of the day, whether you look at this example for the manufacturing manual for cars, or manufacturing manual for laptops, or manufacturing manuals for, I don't know, TVs, or for radios, or for air conditioners, etc. Once that the product at the end of the day is in the same category, okay, is in the same, in the sense of biology, in the same phylogenetic category, okay, then you will expect, because if you're looking at TVs, or if you look at electronic equipment, now if you go to the larger category, you know that all electronic equipment will have capacitors, resistors, coils, wires, printed circuit boards, etc., etc., fans, power supplies, etc., etc., etc. So all those parts regarding you are building a TV, or you are building a laptop, or you are building a mobile phone, for example, will be very similar. The instructions for creating the parts. And then when you go into the assembly instructions, then they will start getting different. When you look at the final configuration instructions, they will be different. But at the end of the day, the part that is different is the part that addresses the instructions of how to put it together. And the power of that is what the example that I have just given in the last question. You can have the two books 100% similar. Mark my words, 100% similar, like the cell in your brain and the cell in your liver. It's 100% similar, but the result is completely different. You cannot, if you look at a brain cell, at a neuron, for example, and a muscle cell, if you have no idea about the genetic system, you cannot guess that those two cells, if you have them, each one of them in a test tube, you will think that each one of them is a completely different creature. Okay? They don't look anything like each other. But what is really making them different is not the content of the DNA, it is the instructions that control the manifestation of the information that is in the DNA. So the point is now, whether the similarity between human and chimp, or human and banana is 50% or 70% or 90% or 98 or 99 or even 100%, it says nothing at all. The question will then be, how using two very similar, or just similar, or not so similar sets of DNA instructions that at the end are ATCG, okay, how can you assemble or create this body of a human, or this body of an ape, or this body of a banana, okay, and they look different completely in terms of shape and function and capabilities. And even if you take the case of the human, you have all the higher functions that are not there in any animal. And the answer will be by design. The answer will be that I can give you the same set of ICs and coils and capacitors and resistors and wires and printed circuit boards and power supplies and etc., etc., etc. But I give you different design, and according to the design, this one set of components will produce a laptop. This other set of components will produce an FM radio. Why? Because the design element is different. Now we go back to the same question. Can the instructions for assembly of design be the product of randomness? The answer is definitely no. Can they be the result of random events being acted upon by natural selection? The result is definitely no. And if somebody would like to claim that this is possible, please show me. When your proposition to the solution of a problem is absurd, you need very strong evidence, and the evidence has to be empirical. Show me. You're saying you're a scientist. You're saying this. Show me. Now, the fact that now, so this is for the beginner. I'm saying that the percentage of similarity is completely irrelevant. But now, is it really 98 point something percent? The answer is no, because the human genome is 5% different in length to the chimp genome already. 5% of 3 billion is plenty. So 5% of 3 billion is how much? So this is 5% of a billion is 50 million. Okay, so you have 50 million. So this is 150 million nucleotides. Just this alone. Okay, so this is completely not present in the other side. The number of chromosomes is different, and this is what is addressed in the Adam or Ape video I was talking about. And then there are the way that they will do the similarity is by aligning genes that look like each other together, and then ignoring the pieces that are misaligned and doing extrapolations and trying to align them by force, etc. So the whole process is really flawed, but I really don't care, because as I said in the first and second part of the argument, it is an expectation that if creatures in general, because we know that all eukaryotes, which is all animals and plants, are eukaryotes, okay, they have to look at each other on the point where you go from ground zero, which is raw materials, to building blocks, which are proteins, to protein complexes, etc., 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 and only at the very final level, which has to be a very small set of instructions that will then tell the system how to assemble a creature, where you will find the trick. So let's say that, of course, they like to twist things and say 99% or 99 or something or 98, but it's not. Let's assume that 100% of the human and chimp genome were exactly the same, and there was only this 5% that gave the extra instructions to assemble the human genome in a different way so that a different creature is created. Or even that almost 100% was there. Then you will need to explain to me, okay, how did the instructions to assemble a human become different from the instructions to assemble a chimp? What kind of intelligence can just have this very small level of differences and produce a very different design? And this is definitely not explained by Darwinism. And then the fifth argument I will tell you is that at the end of the day, someone there was a paper, okay, where they compared what's called the proteome. Okay, so the genome means all the collection of genes. The proteome means all the collections of proteins. So they made an inventory for all the proteins in the body of a chimp, and an inventory for all the proteins in the body of a human being. And the result of the study was that the similarity is 20%, and that there is 80% difference between both. And when I said that, and on one scientific forum, somebody else told me that there was another study that was made, and they found that the similarity is 30% or 30% or something, not 20%. And I laughed, because 30% or 20%, what are you talking about? And this is the amazing thing that I'm telling you about, that even just look at your brain and kidney and liver cells and your muscle cells, and it's the 100% the same genes, okay, but they are manifested differently because the epigenetic layer plays a crazy role that we do not grasp 100% yet. And if you go and watch my discussion even with Dr. James Tour, you will find that he was talking also about another level, which is called the interactome, which relates to other arrangements, probably in the cytoplasm of the cell. And those layers, we don't understand anything about them. So at the end of the day, the percentage is irrelevant. Number two, it is expected that the percentage should be high. Number three, please tell me how the design changes can be so compressed this way, and if there is a mechanism to produce those designs by random mutations and natural selection, which there isn't. And at the end of the day, I can tell you that doing further empirical tests proves that the results of transcription of the genome, which is the protein residues, that the proteome that results from it, shows that humans do not really look like apes in terms of their proteins. And then, for example, I think somebody did even a study about humans and bananas, and humans and bananas are both eukaryotic. and there is a big percentage of similarity. Other than that, some humans are they really go bananas. I don't think that there is real similarity between both when you look from the outside. So sometimes similarity is big and on the genetic level, and you don't see the manifestation, but there is wisdom from Allah subhanahu wa ta'ala for this, because since humans and plants are both eukaryotes, it occurs that we can eat plants. It occurs that since humans, plants, and even bacteria are using the same materials and using same design, fundamental design concepts are the same, bacteria can feed on our bodies when we die. And this causes this whole ecosystem of living things to integrate, have symbiotic relationships, and to live together and help each other survive. Because we eat plants, plants eat fertilizers that are caused, for example, by output from cows or sheep, and other animals that die are decomposed by bacteria and they fertilize the soil, etc. So there is this closed loop that enables this integration of the ecosystem. And then the fifth thing is that obviously humans versus chimps, humans have features that are completely unmatched and unparalleled to chimps, completely whatsoever. Another thing is one thing that I mentioned in a debate that I had with an evolutionary biologist. I think it was Dr. Dan Stern, and in that one, where I was discussing novelty. Okay, I think it was this one. Okay, so you can go and check it out. You will find that human genome, okay, has like 1,700 genes that are called orphan genes, that you cannot trace them back evolutionarily to anything, not to apes and not to anything. What does 1,700 genes mean? If you were listening to me earlier, and I was saying that the least or the last universal common ancestor, Luca, is proposed to have had 355 genes only. So 1,700 genes in humans that have no ancestral lineages whatsoever in any other creature, this includes apes, is like as what, 5 times almost as big as what evolutionists claim to be the complete origin of life. And this matter of orphan genes, hence again, defies the proposition that Yoshi has asked about in the beginning, that all genetic material is coming from duplication. Because what it means that this gene is orphan is that you cannot trace it back to a duplication event that then mutated. And this matter of orphan genes is all over the phylogenetic trees. That when there is novelty, then there is this sudden infusion of information, and you cannot trace the novelty back. Of course, they do not talk about those things. They don't talk about those things because it keeps defying the premises, the basic premises of their arguments. But they don't talk about them. They just keep giving you the nonsense of duplication events and automatic transpositions, and followed by mutations, and then followed by natural selection. And then they don't explain to you how novelty can arise from something like this, because they can't. They can't explain how this level of mutation will still make the creature survive, given mutations in regulatory networks that guide the developmental evolutionary process of the embryo, the development of the embryo. If these things get mutations, they are destroyed. They don't tell you or think with you how this can be integrated in the developmental process of the embryo, to the extent that the embryo can actually have those novel functions integrate with the existing functions. All of this is completely not explained. And I would also really suggest that you look at my latest interview also with Dr. Gunter, because he can put your finger on those events in the history of life where there are those continuous jumps of information. And this includes, of course, the human being. Continuous jumps, they're called saltations, continuous infusions of, I talked about it also with Dr. Bihi, continuous infusions of information that come throughout the history of life, and many of them come in sudden bursts. There's a sudden burst of information in the Ediacan, there's a sudden burst of information in the Cambrian, a sudden burst of information in the Ordovician, etc. Mammalian creatures arrive, flowering plants arrive, sexual reproduction arrives, eukaryotes arrive, etc., etc. All of those human beings arrive. All of those events, which are essentially the history of life, cannot be explained by Darwinism and will not be explained by Darwinism for the very simple reason that order does not come from chaos, design only comes from a mind, information only comes from a mind. Information and design are the children of intent, which is the child of purpose, and purpose and intent and information are the products of a mind. And there is a creator for this universe who created everything. So I hope that was informative. Yeah. So Yoshi says, Sabur Ahmed has spoken about orphan genes. I recommend his videos. Yeah, I actually had this interaction with him on the other show. I can't remember the name of the show, and yeah, where I also talked about this orphan gene matter. Yeah, so here you have also Salahuddin says you explained it. Jazakallah khairan. Thank you, brother. So I hope that was helpful for you who have watched this. I guess we are like 5 minutes away from doing an hour. So if you have like one more question or something, we can close the hour or we can call it a day. But I really want to ask you all to share those discussions with people who have, they have similar questions, and also share the evolution course and this series, Seeing God in Biology, with others. Question regarding Harun. I didn't see the question regarding Harun. I'll go and I'll look up. Okay, so Yoshi has a question about a person called Adnan Oktar. Yeah. So I understand he created this big book that criticizes evolution, and that some of the stuff there is are valid arguments. I have issues with the character itself due to other things that he is engaged in that I really cannot explain or understand how the whole character comes together. So I'm skeptical about the whole configuration of things that he represents. Yet what I'm trying to do, as you all see, is to bring people who are really scientists in the area, where we can go to the core knowledge and really find out about things from people who are experts in the field. But I might as well look into other sources. If you can push something at me, I am happy to do so. All right. So Sharif Sakr says, what is your plan in Ramadan for the stream? All right. So I think we have like we are now at episode 3. I think we should be able to complete 10 episodes before Ramadan. If we finish the 10 episodes, then we'll have season 2 after Ramadan. In Ramadan, I'm considering a few things. One suggestion that looks like a good one is to have a series in Ramadan about the Quran, because Ramadan is the month of the Quran, where we might be addressing ayat in the Quran that has scientific implications that are miraculous, in the sense of the Prophet Muhammad, peace be upon him, would have had no idea about those things if he was just a human being. And then the explanation is obviously that he could have only known if he is a prophet. So like signs of prophethood kind of thing in terms of science. So this is what I'm thinking. If you guys have some more ideas about Ramadan, please let me know. 100 books. Wow. Okay, so I'll look at that. So thank you everybody for being here, and I hope you are all getting ready for Ramadan, and it's a great time for self-purification and contemplation and fixing our relation with Allah subhanahu wa ta'ala. So I hope that you will all grab this opportunity, and we will also have an opportunity to have great time together there, inshallah, with both Quran and science. If you have suggestions, send them to me on email or so Sharif says a Quran series will be great. Thank you for that. So thank you for the encouragement indeed. Okay, so we'll be now I'll say now assalamu alaikum wa rahmatullah, and peace be upon you all. Don't forget to share and like this to others, and see you again next Thursday inshallah. And there will be an interview with an important science philosopher on Sunday that I will probably be announcing tomorrow. I'll just close with this final comment from Yoshi. He says, thank you, Ahmad. It was really fruitful as always. Have a nice day. Thank you, my brother. Have a nice day too. Salahuddin says, Assalamu alaikum wa rahmatullah. Assalamu alaikum, everybody. And let's go for the extra. And I see you again next Thursday, inshallah. Assalamu alaikum wa rahmatullahi wa barakatuh.